Here we go. So, hello everybody. I'm really pleased to welcome you to our second speaker session for this ASA webinar. So, ASA stands for the Ecological Society of America. We are a small society that englobes acarologists in North America. We have around 80 to 90 members, and this is our third year where we host an international webinar series. The goal of these webinars is to really share the research of acarologists around the world and exchange ideas, exchange research so that we are aware about other people's research around the world. Today, I'm really pleased to have Enrico de Lillo, a full professor at the University of Bari in Italy, speaking today for us about Eriophyoids. And Leslie, who helped me organize this webinar, is going to introduce our speaker a little bit more. So Leslie, the floor is yours.
Thank you, Emily. Super happy to have Dr. Lillo with us today. And he got his master in agricultural science in 1985 at the University of Bari, and his PhD in agricultural entomology in 1989 at the University of Naples, with his thesis in Eriophyoids. He was a teacher of the Acarology summer program in Arkansas in 2025. And currently, he's a full professor of general and applied entomology at the University of Bari. He has a great trajectory, and he has published more than 210 full extended papers in national and international journals, with more than 17 citations and an index of 21. His research, sorry, I forgot to mention, he also has published 5 books, chapters in books. His research focused in functional morphology of the mouthparts of phytophagous, predators, and parasitic mites, and of the cincilla to the mouthparts of the same species on legs and prodorsum of pygmy mites. About Eriophyoids, studies that he has done involve systematic geographical distribution and biology, a rigid method for collecting their saliva secretion to be applied in biological and chemical assays. About 80 new Eriophyoid species has been described, and with particularly to candidates in biological control of weeds. And also his research focused in bioecology and control of Colomerus vitis on grapevine. And thank you, Dr. DeLillo, for come and give this seminar to us. It's now
you are welcome.
I leave it to you to take us to your great presentation.
Okay, so I can start.
Yes, the floor is yours. And just maybe a reminder to mute yourself, please, all the audience, to make sure there is no interference with the noise. Thank you.
Okay, thanks for your presentation. Thanks for your invitation. A good morning to all of you. I should say buon pomeriggio in Italian because now it's 5 o'clock here. It's a pleasure to share my knowledge on this taxon, and I hope you can forgive me for my imperfect English or American, as you like. I will try to do my best during my presentation. This is just a summary of what I'm going to discuss with you. There are a few information introducing the taxon and some aspect very intriguing on their biology and ecology. First of all, we have to say that Eriophyoid mites are really tiny and rarely they can be seen by naked eyes. This makes hard working with these mites, of course. What happened about the color? The color is very similar to the plant structures, like trichomes or plant surfaces, and this make much more hard working with them. Usually what happens that the vagrant species are colored orange or reddish, whereas the other species usually are whitish or yellowish. At the same time, also the shape can appear differently, and you can have Eriophid mites with so different shapes, like you can see in this slide. You can have fusiform or spindle-shaped, as you like to say, or cylindrical or worm-like, and sometimes they can be really dorsoventrally depressed. But you have to pay particular attention, care about these shapes, because they can sometimes cause some troubles in the identification of this mite. And we will see a few examples later. In addition, these shapes is also associated to an annular organization of the body, and also on the presence of just two pairs of legs. All these characters favor the access of these mites into small spaces, and also their movement can be easier in very complicated plant structures like buds or witches' brooms or something else. So, and you have to consider also the presence of the different annuli or semi-annuli. It depends on the shape, on the size that they can have. About the mouth parts. The mouth parts arrangement is very peculiar and really unique, unusual among all the acari, all the mites. The mouth parts of the Eriophyid mites are provided with 9 minute stylets. According to someone, they are 7, but this aspect needs to be much more clarified. The cheliceral digits, yellow in these pictures, in these drawings, are modified in thin, very thin stylets, and each pulp develops two stylets in this case, the green parts in this picture, and they are stouter and wider than the cheliceral stylets. Also the labrum, the red one that you can see in the picture, is elongated and quite styliform. In addition, a particular structure, the motivator, which is quite green at the base of all the structure, pivots all the stylet complex, usually the cheliceral stylets, to be honest, giving assistance in their penetration into the plant walls. What about the reproduction? The reproduction of this mite does not require the mating. Males do not mate with the females. They are spreading sperm masses on stalks, producing the so-called spermatophore. And you can see in the picture some of them, and also the ventral side of the male in the coxal genital region. This spermatophore can be sterile if do not have a sperm mass, or fertile if when the sperm mass is present. In this case, in the center, you have a stalk without the sperm mass, whereas on the left of the pictures, you can see the stalk, the head of the stalk, and also many sperms without any flagellum. The female is provided with a cover flap, like you can see here, a sort of door that the female can use for catching the sperm mass and introducing only the sperm mass into the spermatice. Usually what happens that just one sperm mass is collected, and one spermatice is used. This is not the rule, but what is usually happen and was studied deeply by Mikalska, by George Oldfield a time ago. About the eggs. The eggs are quite large in respect to the body of these mites, but they are deformable at the laying and can pass through the smaller genital opening without damaging it. But what happens in the aged females? That the muscles of the genital area can be less efficient, and in some cases the eggs remain inside the body and complete the embryonic development. We don't know, and this is an aspect that should be studied, what happened in the hatched larva inside the body of the mother. The mother for sure died, but what happened for that larva? Is able to come out? This is not known substantially. About the reproduction, we have to say also that we are talking of haplodiploid mites, and in which we have males haploid and the females diploid. And this is quite interesting from a particular point of view. This is a condition very useful when a female reaches a place where no males are present, and these females was not able to get sperms. What happened in this case? That the female can lay parthenogenetic eggs, wait for the maturity of the sons, get the sperm of her sons, and can produce in this way diploid eggs. So you can imagine that the inbreeding is quite usual, and often the sex ratio is quite in favor of the females, taking in account that usually the female is able to lay about 50 eggs, and a few of them can produce males. Concerning the ontogenetic development, this is quite simple. From the egg hatches a larva, which can molt in a nymph, and no more juveniles are known. Usually, juveniles resemble the adults, but there are a few cases in which the difference can be more evident. And this can be quite strange in a case that we have recently studied and regard Phytoptus avellane. Phytoptus avellane is the big bad mite of the hazelnut, or filbert, if we use the English or the American name. And in that case, it was well known the presence of a very particular juvenile that was indicated like Tegonotus-like nymph. And you can see in the first picture on the upper part the molting larva, in which is possible to see a different shape of the body of the inner juvenile. And then you can have on the left, in the part down in the slide, the shape that this mite can have. It's quite depressed dorsoventrally. It can present some lobes on the lateral side. There are few dorsal semiannuli. And then from this, on this picture, on the upper side, on your right, you can see the female coming out from the exuvia of this stage. This is the nymphal stage of indicated like Tegonocus-like nymph. But we can have also a vermiform-like nymph. At the beginning, it was considered like a sort of variation, but studies made in Turkey and at the same time in Serbia demonstrated that these two morphs belonging to different species, Actually, we were able also to observe the differences between the adults, but it was quite hard to find them because they are very, very small differences between females of the Techonotus-like nymph pattern and the vermiform-like pattern. The differences are present. These data are not published yet. We are working on that paper, but it is quite interesting and indicate the need to study all morphological morph and all character, I mean, and also try to get much more information as possible, especially during long period of the time. I will show you another example about this. Eriophyid mite can also have a seasonal polymorphism. This polymorphism depends on the chance that these mites can have to overwinter in a more adapted morph, suitable to go over some stresses that can be during the winter. The polymorphic female can be morphologically evident or not. I have to say that there are few species for which we know also a polymorphic male. These polymorphic female are known for a large number of species, but not for all of them. And the biological advantage of an overwintering morph is its physiological adaptation to the winter stress. And so we have a spring atom morph that we call protogyne and a winter one that we call deutogyne. Sometimes the differences between these two morphs are quite evident. Look at the Calepitrimerus vitis. We have the protogyne on the left and the deutogyne on the right. But also the case of Aculus thaiangensis presently studied. And in this case, we have also very different shape of the prodorsal shield. And for some extent, also for Acheria masalongoi, we were able to find just a very little difference concerning the posterior part of the opistosoma, the posterior part of the dorsal semiannually. Sometimes it's not possible to have this kind of observation and this kind of data, and this is the case of Colomerus vitis. For this species, we were able to find very, very, very few characters able to distinguish and separate the protogyne from the deutogyne, the protogyne on the left, the deutogyne on the right. And regarded the presence or absence of microtubercles on the posterior dorsal semiannuli and the length or some setae at the level of the legs. Why and how we were able to get this information? This is quite intriguing aspect because it opens a way, an approach on the research that we applied. Actually, we moved in this way. We collected more times from the same vines during 13, 14 months the material. So we collected buds and we collected also Erinia because Colomerus vitis it's able to produce Erinia on the leaves of grape and not only on the leaves, to be honest. So in the same area, on the same plants, we collected periodically 2 times in winter and 1, 2, 3, 4 times in the other season, the material. And we studied, at that time, we studied 17 morphological characters. We selected these characters because they were able to be measured with a certain precision. And after that, we also applied the sequencing of some areas of the of in this case, ETS1. We worked on that. And we did this for the mite collected from the Erinia and the mite collected from the buds. Here there is one aspect that has to be considered and underlined. And so, how many mites have to be used when we are working on the DNA, mitochondrial or not mitochondrial? Some people work with pools, but usually it's better to work with a single mite. It seems to be quite difficult because they are so small. Some people try to use also small material for like a hammer for breaking them. But sometimes this is not useful and is not needed. It is enough to use Calyx, which is a product that can break the integument of the mite and can get in this way a lot amount of the DNA. Other people are used to use different material able to preserve the integument, but in this case the success is quite low. Probably is better to combine the two techniques, and so to apply the method which can be more conservative for the integument, and at the same time for other specimens to apply a more destructive method materials, and then compare the sequences that can be done. Why in this way? Because in this way it's possible to have the integument, it's possible to have also the morphological aspect of the mite on which is possible to work on. And this is because it's not so common, but it happens. We can have mixed population. I mean, population composed by more than one species, and this can cause some troubles. This is the case of Toptus avellane, for example. I show you that before. While we worked and which was the advantage that we got from a research point of view working on this way, in this way on Colomerus vitis. The difference is that we got allowed to clarify the annual life cycle of this mite and establish when the deutogyne started to be produced and moved to the overwintering sites. That was great because we started again to examine the slides. We prepared the slides with more than 100 specimens. And so we were able to establish the percentages of the different morphs during the year. And we were able to start to see when the deutogyne started to appear. This can be very useful, especially when you have to establish a management strategy of some mites. In this case, can be useful to establish when it's possible to apply chemicals or other strategies on the individuals, on the population, before these will be closed into the buds. Which are the host plant? The host plants of Eriophyte mites belong to the vascular plants, and currently are known a few species collected from ferns. This because they are not studied too much, but it's possible to get much more information. And about 80% of Eriophyte mites appear to be species specific. In other words, they are monophagous, and many other species appear to be associated to a narrow host range, usually belonging to the same host genus plant and belonging to the same host genus. However, we have to take in consideration some aspects that can influence our results. First, the most of the species is considered monophagous, okay, but this is just based on the collection data. And any assays have been carried out in order to verify the real host range. I mean, host specificity tests. Second, there are only collection data without description often, or with very short ones for some oligophagous or polyphagous species. And this is another problem. And often new host records came from the collection of a very few individuals. This can be the consequence of the wind dispersal. We have to take this in mind. And some records could be just occasional records. They must be verified. The last, in many cases, the description of the same species found on different host plant species, also phylogenetically close one to each to the other, are not completely matching among them, among this morphological description. This makes possible to advance the suspicions that they are sibling or cryptic species, and the current available morphological study appear to be not enough to clearly separate them. There are many cases from this point of view. Which is the relevance of the Eriophid mites? And we have practically four chances. The first one, like pests, specialized and obligate plant feeders. They are obligate plant feeders and can have a direct impact on crops, ornamental plants, urban and forestry plants. There are many examples from this point of view. And the infested organs are usually, or I have to say, the green one. There is no infestation reported, of course, on roots, even though sometimes some of them can be found in seeds, but in the cover parts of the seeds. So they, based on the short and very tiny stylets, can feed on thin wall cells. So they need young tissues on which they can feed on, or tissues that can be really young, even though they can be found on aged part of the plant. So soft organs are needed. And in this case, we have mainly biochemical effects than mechanical effects. Many of them have been found vectors of plant pathogens, in particular viruses, but some of them have been used like biological control agents or weeds and infesting plants. And often they can be the prey, mainly of the generalist predators. Usually, we can classify them in two ecological classes. Vagrants, said also free-living mites, usually they have a fusiform or spindle-form shape, and dorsally they are quite smooth, mainly belonging to the Diptyliomyopide, some in Phytoptide and others in Eriophide. I'm using here the classical classification in Diptyliomyopide, Phytoptide, and Eriophide, even though a new classification has been proposed and consider a different arrangement of the families. But I'm not using that in this case, just for giving a more didactic representation. Then we have also hidden living mites, so-called hidden living mites. They live mainly in natural shelters that can be buds, leaf sheets, or other parts of the leaves or of the plants. And many of them can then induce deformities of the plants and the galls. They are also said gall-making mites. And a few of them can also live under the plant epidermis and cause in this way blistering of the plant, mainly on the leaves. Here there is some example just to show you what can happen. This is the case of Cecidiophysis andersoni, which is a vagrant on yucca, but also Acheria ficus, which can transmit a virus and can be the cause also of a mosaic by its feeding activity. Or, for example, in this case, a silvering produced by Aculus phocae on peach. Or we can have a rusting produced by Aculops lycopersici on tomato. And this is a initial symptom, but then this mite can cause very, very strong deformation of the plant, and also the plant can die completely, or can show some bronzing on the stems, on the fruits, with the cracks that we can see also on the fruits. And then we can have also the coloration on the leaves, like produced by Callipytrimerus vitis, or also the formation or the formation of the canes, short internodes, and also reduced grape production, like in Callipytrimerus vitis. Or we can have the big bud produced by Phytoptus avellane, buds that then die or can produce stems which are not good in developing. And also the same at box 3 by Eriophyoids canestrini. Again, the buds are completely deformed, they became the galls. And also fruit galls, the well-known Acheria sheldoni on lemon. You have to take in mind that just one mite is able to produce this kind of deformation. And then we can have stem galls, like that of Acheria caulobia on Sueda vera. And they are very big. They can be more than 2 centimeters large. So it's incredible size of these. Or we can have typical leaf galls on Acheria, on Bacarices salicifolia, on alnus, on maple, etc., etc. These are pouch galls. These on maple, for example, or can have also a different name based on the shape that these galls can have. And then on grapevine, we can have Erinea. This is the typical Erinea on grapevine, on the ventral side of the leaves, but also on the dorsal side, on the dorsal surface, on the upper surface of the leaves. It is possible. These were, of course, obtained in laboratory readings. And then we can have the witches' brooms, the Wirtshopfe, German word that was used on willow, weeping willow, or in Chondrilla juncea, or in hash. So you can have all this kind of deformation produced always by the mites. To be honest, there is a doubt that this kind of deformation could came also by an association with phytoplasmas, but at the moment we don't have any confirmation concerning this. And then we can have the rolling of the leaf lamina on the border of the leaf lamina, like this in pistachia or in pomegranate or in Euonymus that you can see here with deformation. Of course, the impact on the production of these plants is not so relevant, but it depends on the amount of deformation that we can have on the plant. And then we can have the blistering, and this can be found on pear or also on different plants in a few cases. So it's possible to have a cavity under the epidermis of the leaf, and the mites can develop inside this cavity. And then we can have a reduction on the biomass. And this is quite interesting because the Salsola cali is an infesting plant in the USA, and Acheria salsole can reduce the development. In this picture that you can see indicated with the E5, the plant was not infested, while on the left you can see an infested plant in which there is not so development of the green organs. So it's quite interesting, this aspect. It was done in a petition for Acheria salsole to be introduced in the USA many years ago, to be honest, but at the moment we don't have information about this. This was a species that I described in the past. All this deformation came mainly by some characters of these mites, in particular the ability them to store saliva into the glands. This black spot you can see is represent saliva inside the glands, and this material came out from the dorsal side of the labrum between the digiti fixi. Stylets came from digiti fixi and are introduced inside the tissues, like in this case you can see here. This is a quite interesting didactic representation in which you can see how is possible the stylets come into the epidermis, the epidermical cell, and be introduced the saliva represented by this bubble from the impaired salivary gland and the paired salivary glands. And here you can see the bubble produced, the salivary droplet, which is in the dorsal side, of course, because the labrum, the saliva came out, move on the dorsal side, and you will see on the dorsal side of this movie to come out a small bubble. Is coming, up, is out, now is there. This picture were made, and also this movie was made leaving this mite in cedar oil, commonly used for microscopy. And you can see movement of this mite also in Vaseline oil for a long time. And it can be very interesting, especially from a didactic point of view, to show to the students how they work. This production of the saliva is not always during the experiment or the observation. And we have seen that there are some chemicals that can induce this emission, and other chemicals in which this process cannot happen. And we have seen also that some neurotransmitters, some chemicals that are used, like dopamine or adrenaline, noradrenaline, if mixed with the medium, can induce this effect. This is quite interesting research that could be done in the next. How these mites are distributed on the plant. The distribution of the plant is quite depending on the host plant. I show you a few examples. This is the case of grapevine. And we studied for a long time Calepitrimerus vitis, and in organic vineyard, it was possible to follow well this mite. And we collected the leaves from the apex up to 9-10 nodes from the apex. And we found the higher percentage of mites at the level of the 9-10 on the leaves of the 9-10th node. So we had this kind of distribution, about 35% there, less than 30, 7, 8, 20, 5, 6, and so on. So there is a distribution like a Christmas tree, for example, enlarging toward the base. When we studied this on a fig into years, we found a different distribution. And we observed that this distribution could change also during the year. At the beginning, we found much more, beginning of the year in June, we found more mites at the level of the first leaves. Then later in August, when it's hotter and the plant starts to produce less new leaves, we found more mites at the level of the third and the fourth leaves. whereas mites were not found on other leaves of the plant. And so this depends on the distribution of the trichomes on the fig. And also concerning, for example, tomato, the percentage of Aculops lycopersici on leaves of tomato plants, we found also in this case when the plants were growing up like in this way, it was possible to find the highest percentage of these mites on the lower part, less on the middle, and more or less on the higher part. But we found mites more on the dorsal side of the leaves and also on the basal part of the leaves. So this can influence the way in which we have to monitor these mites, sample these mites, especially when we are, for example, working or carry out experiments for the comparative evaluation of different chemicals or different strategic methods. So we have to take in consideration this aspect. How they disperse. This is a big aspect, quite interesting and important, very important. This is a question that the people working in the weed control often ask to me because, they would like to know where the mites have to be released and if they can reach some places that are not considered and are not in their mind. These Eriophyoid mites move through the wind. They are dispersed. Also, the propagation materials can disperse them, especially when these mites are inside the buds or some recovery that we cannot see or we are not inspecting. But also worker clothes, agricultural tools, and a few other means can be used. I am always very curious, and my curiosity came from this picture. This picture regards Oxygenus mites, more probably, Etegonotus depressus, probably, that was found in the nose of a person that was sent to me by a dermatologist. I am always convinced that if I go in a crop which is really infested by Eriophyoid mites, and I stay there for half an hour and came out and used to recover the mucosa of the mucus of my nose, I'm sure that I will find a lot of Eriophyoid mites inside. And often, probably, what happened that sometime we start to have HU. I don't know in English the name. I started to have this kind of reaction, and probably they are caused by Eriophyoid mites in that condition. They are kept and catched by the mucus of my nose. Why is it quite interesting, this aspect? This aspect is quite interesting for many reasons. In which way they move? There are some observations made by different people, by Mikalska, for example, or Anna Skorachka and her group also. And other people worked on this topic. Also, American people worked on this topic in the past, and not only Polish people. And what they see that this mite can erect on the anal lobes and move the legs like a sort of I don't know, a crawling action to keep the best wind for them. Sometimes they can concentrate in the most exposed areas on the plants, like Tetranicus, like spider mites, or they can form chains like acrobata in some circus, or they can change the position of the body. On the leaves. For example, people in Poland studied Acheria tulipe and Tossichella, and they saw that when the wind is too much, they don't keep this position, but they start to be much more parallel on the leaf surface because the wind is too strong for them. They will lose the chance to be dispersed. But they have also the chance to be very resistant to the stress. We tried some experience with these three species, with Phytoptus avellane, Aceria caulobia, and Cecidophyopsis andersoni. And we left them at 5 degrees more or less in Vaseline oil and in water, adding an antibiotic. And we checked how many individuals, specimens, were died or were still alive. And we saw that in the case of Phytoptus avellane, they were in Vaseline oil alive up to 5 weeks. This in Vaseline oil. And in water plus antibiotic, we had 7 weeks, 6, 7 weeks. So it means that they can stay in the cloud. This is our idea for very long time, especially when the cloud are close to the atmospheric zero. And this experiment was done also with the Colomberus vitis, working with the deutogynes and the protogynes. And we got a very interesting information concerning the resistance of the deutogynes. The deutogynes are reported in green, the protogynes are in red. At 5 degrees in water, they were able to survive for 6 weeks. In Vaseline, we had the same result. Whereas in the protogyne were not so strong. And also the deutogynes at 25 degrees, they were able to resist for 7 days or 10 days. This means that these mites can have a variability to stay without food for so long time and surviving in this stressing condition. So they can be moved passing to the to cloud on our idea, or and move for long distances, or can be moved short distances to the plants which are very close to the starting point. And we have the idea that they could work in this way. I mean, in the lifting, in the taking off, they can utilize the thermal current for reaching the cloud. Why the cloud? The cloud, because in this way they can keep the water of the body. They are not dehydrated. And then they can descend using the body, like in the picture that you can see on the right. So in that way, and this can be possible. Why? Because at the base of some setae we can find the muscles. This is a C2 setae of Phytoptus avellane on the dorsal side of the body, and at the base of this tubercle, setal tubercle, there are muscles that can change the position of this setae. But at the same time, also the legs. These legs can move differently, like this structure that you can see here that can be moved by the operator. And at the same time, also the body. You can see these bold areas, bold lines are muscles. So it means that the body can change its shape or can change its position in different way. It should be great to find someone able to use modeling for trying to apply some ideas and try to verify if actually with the current model concerning air pollen or something else is possible to demonstrate this theory. Finally, how we can catch Eriophil mites. This is the water pans, classical water pans used also by Amrin student Zhao, Shifu Zhao, that was able to collect a lot of Eriophid mites in this way. But the problem is that you have to go every day to collect material. We invented this kind of structure in which it's possible to collect Eriophid mites once per week. This is a prototype, it's not published. We have just prepared the documentation for a patent with an evolution of this prototype. You can understand that I cannot show you that structure because it's a patent that we would like to get. And here you can see how it works and what we were able to get. We were able to get easily Eriophid mites using this kind of system with the reservoir of the fluid, with a liquid containing detergent and also an antimycotic material. And then we have the chance to collect the excess of water caused by rain or something else. So I have finished, and I believe that I am on time. And I would like to show you some of open questions, other those that I have showed you that can be intriguing for further research. For example, we don't have currently an artificial substrates on which rear Eriophid mites. There is just one case reported in literature quite recently. We don't know how long they can be dispersed by wind, and you have seen the aspects concerning that. We need to consider better the cryptic species. Probably there are many are so. For example, someone was publishing something about Aculus phocaevi, distinguishing different species. We don't know almost nothing about the chemical ecology of Eriophid mites, pheromones, semiochemicals, etc., involving these mites. How long Eriophid mites are able to live on an occasional host. This is quite interesting aspect, which is quite relevant in the case of the weed control. And it's possible to replace hydrate chloralium in long-lasting like mousting, getting a good result. We don't know. We are looking for transcriptomic viruses, etc., etc., etc. You can see here a long list concerning this chance. So I have finished probably 5 minutes before the time that was established, and I hope that there could be some questions, and I hope also that this presentation was interesting and added to you new information.
Thank you, Enrico. Oh, what a sweet dog. Is it your dog?
Yes, it's my dog.
Really nice presentation. I'm gonna let the audience chime in and come up for questions. So if you have questions, feel free to write it down in the chat or unmute yourself. Matt Burton say, how many Eriophyidae on that dog? Probably a lot. I have a question concerning
Maybe there could be many area on the dog. I'm looking
the chat.
The chat, yeah.
Had a question regarding your water trap. That is really interesting. So you want to commercialize that for monitoring Eriophyidae population or to control, to do mass trapping and maybe combine that with an insecticide or bait or something that can kill?
Yeah, I understand. No, I tell you, we have just presented a project concerning the application of this trap. And there are some cases in which monitoring is crucial. I mean, you need to have information concerning the presence of the mites and that mites reach the crop time before the appearance of the symptoms. This is the case of Aculopsilico persici, the tomato rust mite. Tomato rust mite usually can show symptoms 3, 4, 5, 6 weeks after the infestation. When you see the symptoms, it's too late for doing an efficient control. So in our case, the monitoring is much more important because it can help a lot. Taking in mind that those mites move mainly through the wind dispersal and not by the plants that you are transplanting.
Thank you. I see. That makes total sense. So we have a couple of questions, so I can go ahead and read them. Mite belongs to the class Arachnidae, and it's an important character of this class that they have 4 pair of legs. So how
I have seen Samuel. Probably Samuel can explain better than me. Samuel Bolton.
About the legs?
Is there? Yeah. Samuel is there. He worked a lot on this aspect, so I believe that Samuel can give an explanation better than me because he related this mite to Nemalticide and gave some information about this. I suggest to talk about Samuel. I am much more an applied acarologist than interested in evolution. So Samuel is the best for explaining and replying to this. Are there many young scientists working on the taxonomy of these mites, both morphology, molecular, and host-plant connection? Unfortunately, no. Arash is there. He cannot. He sent me a message. Arash Onarmand is one of the youngest. There are very few new scientists, young scientists. It is important to have more and more and more. A second question is that how you capture such amazing photos of these tiny creatures? What do you mean? Do you refer to the scanning electron microscope or to the micrographs? Scanning electron micrograph, I got many of these pictures in the past when it was possible to utilize a microscope, a scanning electron microscope that was easy to manage. That if you ask to someone, to operator, to do what we did, that operator will kill you, because we took the mites and we put the mites in a small drop of water, distilled water. Then we sucked the distilled water. The mite remained stub, and we put that in the column, and we were able to keep the picture without preparing the mites, without doing anything. Now we don't have anymore that scanning electron microscope, so it's not possible to do that. For the picture that we got to the light microscopy, we are using an Eriophyoid mites a microscope, a BX50 Olympus microscope, and we mounted on that a Mark III Canon camera. It works very well, and it's possible to get very good results on the slides, on the mounted slides.
There's a lot of more questions, but one is about the single species have 2 forms of a body. Is it possible? I guess you answered this question in your talk, kind of.
No.
No. Okay.
No. If you to morph, you mean to shape of body. Based on my experience, I don't have knowledge about this. I didn't see. Is there some species or genera be erected based on the season polymorphism variation shape of body? Is there some species or genera? No, you cannot establish a species or genera on the deutogyne. You have to do that on the protogyne. In fact, some changes were done in recent years in which it was possible to distinguish that. One of the aspects that is negative, and I have to say, is that some descriptions are made just on females without finding the male, without describing the male, or without indicating the presence of juvenile. Arash knows that very well because he was in my lab and I was very oppressive about this. It is important to have also males and juveniles to say that that one is the right female for that species and not a deutogyne. So in the past it was not so. Many have different morphology between the, is that what you mean? Or are you asking about phenotypic plasticity? It's possible to have a phenotypic plasticity, but from a morphometric point of view, not large variation concerning the morphology.
Okay, we're going to wrap up with one more question, which is really interesting. Are they updated taxonomic keys one that we can use?
The most updated key was published by Chandrapatia and Amrin, but it was not complete. In the catalog of Thailand Eriophyoids. So you have to use always Amrin and Stasny key, and you have to add information about that. I'm trying to put together, but it's quite hard to do for academic questions. I mean, I am too much involved with teaching and other stuff, and preparing a key requires time. If anyone is interested, I enjoy the challenge of taking images of these mites and can share my techniques and equipment if people want. Thank you. Thank you, Matt.
A lot of cool picture. Unfortunately, I have to be respectful of everyone's time, and I will have to wrap up the discussion session. Maybe, Dr. De Lillo, can I put your email maybe in the chat?
Yeah, of course.
To follow up with question that way. So I'm going to share Dr. De Lillo email. Here it is. You're welcome to ask him a question individually. Thank you so much, Dr. De Lillo, for your time. It was a really great presentation. Again, we will send the recording of this video to our members, and we'll also post it on our YouTube channel. So if you know friends, colleagues who wants to watch the video again, that will be available on our Ecological Society YouTube channel. And I'm sharing the channel here.
Can add just one thing, Emily.
Yes.
Thanks to all of you. I would like just to say to all people that it was just a way to introduce you to the Eriophyoidea.
Of course.
It's quite, there are so many things on which we can discuss, and I tried to select the things that were the base for this topic and to give you the chance to have some ideas for the rations. For example, one stupid thing is the question how they are able to move on the lower part of the leaves without falling down.
Yeah. Yes.
So the questions are so many.
And you're welcome to come back anytime in the next year if we have more webinar. That would be, you can dig into more topic if you want to. We'll be really happy to welcome you again.
Okay.
So, again, thank you so much for your time. I wish you a nice afternoon and a nice day for the people here in the US. And well, have a nice day again. Bye.
Bye-bye.